So thank you for coming here. See a lot of different people. That is really nice. And we're pleased to have Douw here. She's an associate professor at the Department of Anatomy and Neurosciences at Amsterdam UMC and head of Multiscale Network Neurosciences there. So yeah, really, we're looking forward to your talk. Multiscale Network Neuroscience.
Great. Thanks for the introduction, Martin, for the invitation. I always really like talking to a diverse set of people, which I think you are, because you all have an interest in complex systems. I work mainly on the clinical side, so my talk today will be mainly on the clinical stuff. I hope to spark a little bit of your interest in maybe working on the brain as well as yeah, it's a little bit light. Can you see it still? Yeah, you can. So I work on multiscale network neuroscience, which means basically what we try to do is depicted by this piece of art on the left by the Langema, where you see all these different plateaus and stairs, and in the end there's a common space in the middle, sort of atrium, where all these different perspectives meet, or all these different scales, and that's kind of also how they see what it is that we do, trying to combine scales. And I work in a very specific group of patients for that, which is patients with glioma mostly. Glioma is one of the most frequently occurring primary brain tumors, but it's still very rare. So there's about 6 annual cases per 100,000. Means that in the Netherlands there are 1,000 new patients per year. There are other primary brain tumors that you might have heard about. So meningioma is one of them. Metastases are, so that's well expected from somewhere else in the body. That metastasize into the brain. That's not what I mean with primary brain tumors. But glioma really integrates into the brain tissue and has this bidirectional relationship with the functioning of the normal brain. Unfortunately, the prognosis for the patient is really bad. So the most common form of glioma is glioblastoma. It's a grade 4 tumor, and these patients have a median survival of about 14 months. So it's not a positive outlook for these patients.
So after treatment?
That's from the diagnosis, and within those 14 months they get resection, chemotherapy, radiation combined, and then chemotherapy again.
And that was 14 months.
Yeah. And go with some variation. So there's definitely people who live a lot longer in the village. Yeah. What we know is that a lot of these patients have cognitive dysfunction, and this is what actually brought me through this whole field and open network science. I'm a clinical neuropsychologist by training, so I'm really interested in how the brain governs behavior, especially cognitive behavior, so how people can do the things that they do in daily life. Lots of these patients have cognitive dysfunctions. They are not able to perform tasks in daily life or think well. But it also offers us a unique chance to do multi-scale research because we have data from both imaging and measurements that we can do in any living human. But we also have tissue, which is kind of the star-like cell that is in the middle of the slide, because patients undergo resective surgery. So we take the tissue of the brain, including the tumor and some of the surrounding tissue, and we can investigate that as well. So that's a very unique window opportunity into that. So the topic of today, I guess, was a little bit of resilience. That's the question I sort of got. I want to underline that although patients have cognitive dysfunction, and this is a disease that has a very dismal prognosis, there's still a lot of resilience. So this is a meta-analysis of all the cognitive deficits in these patients. So it takes all of the studies that have been done in glioma patients at diagnosis and looks at how many of them have cognitive impairments. And you see the percentages on the y-axis. So if you look at any domain, which is over here, you see that almost all patients have some sort of cognitive deficit. The median is about 60%. But then if you look at the different domains, these numbers vary a little bit. So a lot of patients have cognitive deficits. And just to illustrate something that I will get back to is executive functioning. It's kind of all over the place in this graph. But we see it as one of the most often impaired domains in these patients. Does anyone know what executive function is? Want to guess?
performing the task.
That's a very general explanation. Within neuropsychology, we see it as the most complex cognitive behavior. So it's a combination of planning, receiving input, processing it, and then generating an output.
So is that similar to the
It has overlap, so executive functioning would be more the neuropsychology term for it. Clinically, you would call it more like cognitive control, which is something that is impaired in depression and a lot of other psychiatric issues, because that also has to do with planning. So it's kind of working memory, which is keeping things into your short-term memory and then being able to manipulate your surroundings and not having that knowledge in your head. Planning is one that's very similar to that, but on the longer scale. And another one that I'm really interested in is flexibility or set shifting, which means that you can flexibly change the rules of what you're doing based on the demands of the environment. And as an example, this is a very simple test. It's called the set shifting test. You see these circles with numbers and letters in them. Patients basically first have to just cross all the numbers separately, then all the letters separately, and then they have to switch between the two. So first a number, then a letter, then a number, then a letter, keeping both in the right order. So this is an example of what they would do when A to B. So they have to switch between rules, letters, numbers, and keep that in there, in the back of their mind. And we typically measure reaction time on this type of test, thinking that if you take longer to do this in comparison to the easier conditions where you just cross out the letters or numbers, you have an impairment or technique. So yeah, these numbers are not great, the percentage impaired patients, but when I sit across from a single patient like this woman, there's no real telling of how she will perform on this test. We might have a scan that looks like this. This is an actual brain tumor. It's actually not a glioma like I talked about before, but a meningioma. It's huge, obviously. And you might think this is so big, this patient will not be able to perform any task. But this could be the patient sitting there, having no issues. So there's no linkage between the size of the tumor, also the location of the tumor, and how the patient really behaves cognitively. This is something that has been called lesion symptom mapping. So basically you're trying to say, yeah, the brain is kind of like a map. And if you have a problem in a specific point in the map, then you have a specific problem. It's also what some people would call globology. So that's fMRI research, where you do a task in a scanner and then see which parts of the brain light up. This is kind of post-process image like that. And then say that's the region that does that task, as if it's a one-to-one mapping. In general, there's doubts about whether this holds up for the brain for more than just simple motor functioning or visual functioning, which are the most basic features of cognition. But for executive functioning, for instance, we see that this doesn't work, especially in brain tumor patients. So we did a study, but there's many actually, where you see here where the tumors are. So the brighter colors, like here in the temporal lobes, this is all slices to the brain, and this is the bottom. And then you go up to the outside of the brain. And then you see that most of the tumors, the yellow bits, are in the temporal lobes, which are kind of like on the side of your head, which is a very common place for a tumor to be. So these are the tumor regions, and you can, at the group level, map whether having tumor in particular regions of the brain would link to cognitive deficits. So we did that. This is the map that then shows which regions would relate to a cognitive problem. And this is processing speed, which is also part kind of executive functioning. And this is uncorrected, so it's also statistical testing. So this is what might be there, but when you really correct for the number of tests performed, you see nothing lighting up. So we can't really map regions of the brain to cognitive deficits in a one-to-one fashion. So we need a different way. But before I talk about actually looking at networks, I want to show you some more about this cognitive resilience. Because one way that we also test cognitive resilience in these patients is to actually do awake mapping. A lot of brain tumor patients get an awake surgery. So the patient is opened up, sometimes during sleep, sometimes awake already. And then they perform tasks in the operating room, while the surgeon is putting electrodes on the brain tissue and just giving a short pulse, an electric pulse, which kind of transiently shuts down that part of the brain that it touches. So it's kind of like a virtual lesion, a transient lesion. And it's a mimicking of a resection. So obviously in brain tumor surgery, you want to take out as much of the tumor as you can, but it's not, yeah, this kind of looks like a ball, but it actually integrates into the tissue. So sometimes you on the border the tumor and the normal brain, and you don't really know, is this functional tissue or should I take it out to have the best prognosis for the patients? And by testing in the operating room, you can transiently check, is this region safe to remove? So that's basically what you see here. This is the patient. So this patient put the video of the operation on YouTube himself, so you can look it up. The patient is here, obviously. This is the neuropsychologist's arm who is doing the testing. And over here is you won't see any blood or anything, so it's safe, but this is kind of the sheet after which the neurosurgeon is. And they are moving their sort of wand with the stimulation from one place to the next and checking whether the patient is still functioning. You can do many different tasks, not all, so you can't really test executive functioning that well because patients are obviously fixated. Their head is in a sort of ring, so they can move at all. That would be a problem, of course, for brain surgery. They can do language tasks, some memory tasks, motor tasks with their hands and feet. And in this case, this patient is opera singer. So one of his big questions, undergoing surgery was, can I still sing after the surgery? So they had him sing, basically, during the surgery, and then test the different parts of his cortex. And it shows how resilient this functioning is. So a lot of the places where they stimulated, nothing happens. You will see it in a moment. But then there's one spot where something happens. So you can take out a lot with the brain tissue, but not one spot. I'm sure.
Im Schnee, nun ist die Welt so trübe, der Weg gehüllt mit Schnee.
The surgeons are continuously moving.
Well done, Ambrose. Well done. Well done. Well done. Well done. Scary. Well done. This is what we do. It's not going down. Great job. You did it great. Really great. I love your singing.
Try again? Should I start it from the beginning?
Yeah.
Okay, wait.
Yeah.
It repeats automatically.
It goes to the makeup before.
I'm gonna film what you do.
And he's fine again.
Yeah, so basically what you saw is that he was able to sing, then they hit a spot that was somehow involved or crucial for his singing. He stopped, well, he lost tone, the voice. You also, if you looked closely, you might have seen some twitches in his jaw, which means that he was probably also in the area controlling the jaw and the mouth musculature, which of course is very important for singing. Patient was smiling, which is good because you do prep patients a lot for this moment because it's obviously very scary. So not all patients are able to undergo awake craniotomy because you have to be able to accept that you're fixated and cannot really act, but you do have to perform a task basically. And then after 30 seconds or so, he's back to normal and can pick up the singing again. And the surgeon has put a little piece of paper on the regions of the brain that were implied in singing. So that's how awake craniotomy works.
What sort of volume is involved? Cubic millimeters, cubic microns, cubic centimeters?
Could be all of those. So typically they use a burning tool to take away very small bits of the brain. The electrode is in the order of a centimeter that you stimulate. So that's pretty small. Yeah.
I think it's very big.
That's individual cells. Well, you're always at a sort of
Thank you.
Yeah.
You can dissect cells because I thought they don't use microscopes for surgery.
No. So it's
So it's in a millimeter, I guess.
Yeah.
The tissue that they-
centimeter, I would say.
Yeah, exactly.
Yeah. Yeah, so this is great that we have awake craniotomy, but it's obviously not applicable to all patients. It's very time consuming for both the patient and the surgical team. There has to be a lot of expertise. You can't do all of the tests, like I said. It's very unnatural to be lying down, fixated, and have to perform a task. So we are looking to achieve better measurements of cognitive resilience without having to do actual testing with the direct electrical stimulation, it's called. So that's kind of where I want to take this whole talk. How can we do better, basically? And it would be great if it's less invasive, obviously. And to do that, we use network science. So we apply graph theory to the brain. I kind of assume that you are all familiar with graph theory. But I just want to explain how we actually get a graph or a network description of the brain. So on the macroscopic scale, we would divide the brain, which is what you see here. It's kind of a surface rendering of the cortex, so the outside of the brain. We divide it into bits, into parcels. This is usually based on a combination of anatomical information, like what type of cells are in these different brain regions. But nowadays, we also use connectivity patterns. So we look at all the voxels, for instance, or even cells in postmortem tissue and see how canonical connectivity is organized in brain regions. And we say that a region or an atlas region or a parcel is a region if it has largely similar connectivity patterns, in addition to anatomically being very similar.
What is this between people?
Yeah, it's a great question. Pretty variable. Yeah, so I will go to the level of sort of modular structure that's very similar across individuals. Individual parcels are very different. So we use cortically annotated ways to match between people. So the folding, which you don't see in this image, but of course the brain is actually folded, it's not smooth. The foldage is very different across individuals, and that says something about how their morphology is organized at the cellular level. And we have methods to map these foldings and the sort of surface curvatures. So we typically try to make it the same, but there will always be differences. Yeah. Is there another question? So these would be the nodes, but we can, of course, also look at individual neurons. I won't be presenting work on that today necessarily, but there are studies using individual neurons as nodes and then looking at synaptic connections.
So how many nodes are there typically?
Oh, yeah, this is what I was going to say. We typically use about 200. we do multimodal imaging, so we use both resting state fMRI, fusion MRI, and MEG. We'll get into that. And the resolution of these methods is very different. And it's about 200 is a nice intermediate between the spatial resolutions. And it's also where you do find meaningful correlates of cognition. So that helps a lot.
So I thought you were using structural connectivity when you come up with this method. When you say MRI, then it is maybe if they are put together, right?
It's a combination. So the map is actually fixed for like an average brain, which is what I'm showing here. The information from both post-mortem studies where you look at individual cells, functional connectivity and structural connectivity is sort of used to parcellate an average brain into these regions. And then we warp this to the individual patient's brain structure, basically. So it has all the information in it, but we don't do it for each individual. So we basically say we have all the information from sort of healthy cohort. In the literature, they made this atlas. It has all the information. And then we simply warp it to the individual patient's structure.
Atlases, I think there are
Yeah. There are many. Yeah, so you can use, like we use a 200 parcel one, but you can go from like 50 up to 30,000. If you go to the voxel level, which is voxels are the smallest individual units of an MRI, then you can go up to 30,000. Yeah. So those are the nodes. The connections we can look at in different ways. So one way is structural links. This is looks like a bowl of spaghetti. It is my brain. So I'm not sure whether it looks good. You can tell from the number of spaghettis. But this you can do by having a diffusion MRI that measures the diffusion of water molecules along isotropic or anisotropic sort of bundles, because white matter, so that's the anatomical links between brain regions or cells, they are very insulated, so water molecules will move in an anisotropic way if they are limited by a white matter bundle, and if they are not, it's a basic diffusion process, so it's isotropic. And by doing this in all sorts of directions, you can sort of establish whether there is a link, so a spaghetti, and also in which direction it's going. So I think here you see that the blue is up to down, or up, down to up. There's no directionality here. When you see this is the brainstem, so you can see that all these different regions on top are going down in terms of the white matter connections into the spine. Green, I think, is front to back, so in that direction. And you can see that it's these different kind of connections. And typically we take sort of the number of connections, or these spaghetti plots, between different regions, or between different nodes, or we take the weight, the number of the. So that's a sort of a structural representation of the brain network. We can also look at functional links. Here we assume that a synchronous time series in brain activity represents some sort of connectivity. I know that's a big assumption for physicists and mathematicians, but it's all we have basically to assess sort of functional communication. And we can measure it in different ways. So this is an EEG recording of my colleague Bernardo. It measures the differences involved across the different brain locations. This is an MEG system. It's actually one of the only ones in the Netherlands. We have one in Amsterdam, and there's also one in Nijmegen. But this is the only one that uses it on patients. It's a lot like EEG, but it measures the magnetic fields that are created by action potentials of huge numbers of neurons. So it's really at the scale of hundreds of thousands of neurons all firing synchronously. And we have, of course, functional MRI, which has been used the most in the field because everyone has an MRI. And it's very easy to collect this type of data. Do notice the timescales of these different methods. So there's a difference in spatial resolution. EEG, MEG are not so great in terms of spatial resolution. So I would say that you can discern about 150 to 250 individual sources based on EEG, MEG. This is why we use the 200 region parcellation. fMRI has a very high spatial resolution, so here you can go up to 30,000. but the temporal resolution is very low. So these are recordings of 6 and 10 seconds, and you see that there's a very nice spread of frequencies. The sample frequency is about 1,000 hertz, so that's a lot. Here, the sample frequency is maybe 1 hertz, sometimes even less, or even more. So the time scales are completely different, and this time series is 6 minutes of data. So you're looking at different aspects of brain activity. And we typically do this during a no-task resting state. So as sort of, some people are confused by this because they think you have to measure while someone is doing a task, but that proves not to be the case. So originally, this task paradigm that people have used also a lot is based on this idea. And this is for both, but you can do this in EEG and MEG as well. So this is fMRI. Basically, the idea of a task paradigm is that you have someone do a task while you're recording or measuring. And this will modulate the brain activity, which it does. This is the simplest task where you open and close your eyes. So it's a visual task. And the idea is that you subtract these two conditions from each other, so the brain maps of activity, and then you see which region of the brain is responsible for the task. In this case, you see the occipital cortex in the back of the brain light up, which is, of course, where vision is located. So it works really well for things like vision, motor, some memory, language. Not for executive function. It's very hard to find one spot if you subtract a task condition from a resting condition. And there is a lot of information in the resting condition. So it used to be sort of a default contrast for the task-based studies. And we've known now for a couple of decades that there's so much information in this resting state. So even if you tell patients or participants not to do anything specifically, like here, you see that there's a lot of variation in the bold response. Bold is the fMRI response. And you see that this change is as big as what happens during a task. So this is a percentage change during a task. And it's at maximum 5%. So the brain is always on. It's always doing a lot. It's always varying and fluctuating. If you do a very specific, very difficult task, only 5% of the activity changes. So that's small.
Is it not possible to compare in deep sleep, for example?
You can.
And then the percentage will change significantly, or?
Yes, it will be very much more flatlined, but the type of connectivity that you see in the resting state is still there. So even under anesthesia, you can see the same type of connectivity that you also see during a resting state if someone is awake, and also the same connectivity that you see while someone is doing a task. So basically, just to explain what you see here is this is a resting state. Patients are not, or people are not doing anything specifically. They're just thinking about nothing. You see that there's a lot of variation in their brain activity. If you then look at this region in yellow left and correlate this time series to all the other boxes of the brain, you find that the highest correlations will be with the other side motor cortex. So that's the left motor cortex and the right motor cortex. And this is always connected, whether you're using your hand or not. And that was a new finding in like the 90s, that there's this much information in the resting state. In recent years, we've learned even more about this. So this is a study, or a series of studies actually, where they did a lot of scans on a group of 10 people. So they measured, I think, like 10 times a day for weeks. They did resting state and task-based fMRI, and they compared what is mostly contributing to differences in activity. What you see here is that there's a lot of structure at the group level, which means that every human has a typical structure of what the brain network looks like. Then there's individual variation. So me and you are not the same in our connectivity, and this individual differences are size of the effect. But the task and the session, either the individual and the task, those are much lower, which means that it doesn't really matter whether you measure in the morning, in the midnight. It's actually called the midnight scan clock because they did a lot of these scans at midnight, or whether what kind of task you're doing or resting state. So most of the differences in network topology based on resting state of fMRI are based on differences between people, not whether they're doing a task time of day. So that sort of suggests, on the one hand, that this resting state network topology contains a lot of information, but it also means that we can maybe use it in a clinical perspective. Because if you're thinking about patients, we're trying to measure differences between people, right? We're trying to say why some patients have cognitive deficits and others do not. So I think this is very important for the work that we do. What we know about brain networks is that they adhere to all of the organizational principles that you probably know from other systems. So they're small world, they are to some extent scale-free, they have hubs, they have modules. And the modules, I think, have a special place because they're, of course, spatially located in the brain. And they're also giving us a clue about why this lesion symptom mapping doesn't work. So if you think about lesion symptom mapping, you think that one region of the brain is doing one thing and the region next to it is doing another thing. These are the modules or resting state networks, as you call them, that you find if you do a modularity algorithm on the brain network. You see that the modules are very spatially widespread. So for instance, a very important one is the red one, which is here in the medial frontal lobe, the medial parietal lobe, and then big parts of the cortex on the outside. So this is parietal lobe, temporal, frontal again. It also entails the hippocampus, which you may have heard of. It's very important for memory functioning. So this is a network that we typically relate to autobiographical memory, mind wandering, relating things in the present to what happened in the past. And this network is always highly connected to itself. So the regions in the distributed areas of the brain are always connecting a lot to each other, regardless of whether someone is doing a task or is sedated or whatever they're doing. Relating cognition to the DMN or functioning of the DMN has been much more fruitful than linking it to individual brain regions. And the same goes for the frontoparietal network, the FPN. And when we think about the DMN, it's mostly a memory network. The FPN, which is orange, so that's large parts of the frontal regions and the parietal regions, which is why it's called frontoparietal network. It's also called the central executive because it's mostly linked to executive function. And again, it's a distributed network that consists of separate brain regions or nodes. So let's go back to glioma. How can we use all of this? Well, first of all, we've spent a lot of time describing the brain network and relating it to complaints or cognition in these patients. Right when I started, is that focal lesions like a glioma, which look like one thing on a scan, have very distributed network effects. So if you look at the scan of this patient, we see that there's a tumor here in the left hemisphere. And up to then, I think a lot of neurologists or neuropsychologists were thinking, okay, then you will have probably problems with the right hand because that's the area that the tumor is in. But this patient might have had severe executive functioning disturbances that were not relatable to location. And what we saw is that the patient's network didn't just contain a hole, didn't just differ within the hemisphere that contained the tumor, but was even very different on the contralateral side. So you see that the global network topology has changed. And I'm showing you this because it's, I think, most intuitive that connectivity goes down if you have a brain tumor. But what we actually see is the opposite. So if you have a normal brain network, you have this small world organization with high clustering, low path length, high efficiency. In brain tumor patients, we consistently find high local connectivity and clustering coefficients. So regardless of where the tumor is, here we drew it in the sort of central region, but you see an increase in the number of triangles throughout the brain, which does not relate to the Euclidean distance from the tumor. The level at which this clustering is higher than in healthy controls relates to seizure propensity. And we also see a loss of integration which relates to cognitive deficits. Now, the question, of course, is this specific to brain tumor patients? Because we see the same type of correlations between clustering and integration in healthy people. People who have a more efficient brain network also perform better. So this is a very coarse description of what happens in the brain network in disease. We also see this for many other types of neurological brain-related diseases. So it's a very general group level association that still doesn't help us in the original question. How can we predict cognition in individual patients? Because the relationship is weak. So what we've been doing is, first of all, sort of scaling up our integration on the imaging side. So we use multi-layer network approaches to, instead of having a separate MEG, a separate fMRI, separate diffusion MRI, with all these different networks and then combining them statistically, we use multi-layer approaches to combine them or to unify them in one go. And this is work by Lucas and Fernando. We basically have these three different modalities here on the left. The crucial thing is to have the same parcellation in all three modalities so that we have the same nodes across layers. Then we do bespoke analyses for all the different neuroimaging modalities because they, of course, need some different preprocessing. Threshold the matrices to get the same number of connections for each of the layers so that the number of connections and the distribution doesn't drive the multi-layer outcomes. And then based on this super adjacency matrix, we can, of course, extract some sort of measures that we think relates to cognition. And this study, this was in healthy controls. We focused on eigenvector centrality of the frontoparietal network. Like I said, the FPN has been mostly linked to the to executive functioning. And we had many executive functioning tests in these subjects. And we think that centrality tells us something about integration across the different modalities of this network. First of all, we found that it was relevant as an explanation for both biology and cognition. So first, age. We know that age has this, the relationship between brain features and age is usually in a U shape. So for instance, the white matter density, so the number of connections in your brain, it goes up until about 25, then it plateaus, then it goes down again after 50. And that goes for a lot of the features of our brain. It sort of goes up and then down, unfortunately. We see the same relationship for this multi-layer eigenvector centrality measure. It's more, so that sort of establishes some biological relevance of the measure. But we also see that there's a positive correlation between executive functioning, which is on the y-axis, which is corrected for age and sex and educational level, and the centrality of the frontoparietal network. So people with a more central hubby multi-layer FPN typically perform better on an executive functioning task. Yeah.
Does every layer correspond to in that multi-layer representation?
It's a modality of imaging. So it's one fMRI layer, it's one diffusion MRI layer, and then there's 6 MEG layers. And I didn't go into the details, but there you can look at different frequency bands as well. So it had 8 different layers. Yeah, so we see that this is relevant as an explanation of cognition, and it was more explanatory than using single layer network topology or adding them all together in a statistical model in a regression. But of course, we were most interested in does this work in glioma patients? This is work that Marike did in my team. And she again found that this multi-layer centrality of the FPN was relevant. She used only MEG to construct multi-layer eigenvector centrality. And she had longitudinal data. So basically, we measured patients at diagnosis, and then they got treatment, so surgery, chemotherapy, radiotherapy. And we measured them again about 1 year after surgery. And what we observed is that there was a correlation both at T1, so that's at the diagnosis time point, in the same way. People with higher multi-layer eigenvector centrality perform better on an executive functioning task. But when we looked at change scores, and that's what you see in panel B, we saw that patients who had declining executive functioning over time also had decreasing multi-layer eigenvector centrality, showing that these two measures go hand in hand over time. Of course, our hope was that we would be able to predict based on T1. So we would do a multi-layer hypertoxicity calculation before surgery, before all the treatments, and that we would be able to predict which patients were at risk of having cognitive decline. That was not possible using this measure. So we had to keep looking. One question we also wanted to answer is how do these sort of macro scale network properties relate to the underlying cellular structures? There was already a question about what resolution are you looking at? Can you look at these individual cells? We can to some extent. So this is a study where we studied temporal lobe epilepsy patients, slightly different from glioma, but still we have all this information from them. Before surgery, they undergo multimodal imaging. They get a cognitive test, and the surgeon takes out large portions of the brain. And here we look specifically at the part of the default mode network, so the one that is mostly linked to memory in the temporal lobe. It's very schematic here. We took out the same region in all patients, and then looked at the micro cellular level. We looked at the dendritic length. So this is a dendrite of a neuron. And then you see the dendrites are sort of the tree-like structures. This is a brain cell, and we know that the longer these dendritic cells or the spines are, the more complex the cell is. And so hypothetically, the more integrated the cell is within the microcircuits. The same goes for action potential. So you can take out the tissue. My colleagues then run to the lab with the tissue, and then they patch clamp it. So that means that they measure individual action potentials after a stimulation. They can measure the speed with which these action potentials are rising. And again, the faster this action potential rises, the more integrative the neuron is thought to be. So we have this information for all patients. On the macro scale, we measured the multi-layer brain network again. So here we have fMRI and MEG. We looked again at centrality, but then of the default mode network, because that was the region that was taken out during the surgery. And what we saw is that these two went hand in hand. So this is sort of a parallelogram between the different cellular metrics, so the length and the rise speed of the action potentials. And here are the macro scale centrality measures of the region that contains a tumor, but it's taking the entire brain network into account. And we see that they're mostly positively correlated. If we zoom in on the individual data, this is obviously too good to be true. I think there's definitely some overestimation of the size of this effect, but all of them are in the same direction. So I do think that there's a positive correlation. And this also holds up if we do permutations. If we leave one of the patients out, it's of course a very small patient population. Or if we link the cellular properties to all the other regions in the brain that we have the macro scale centrality of, it's still specific to this region. So basically, people that have cells in their temporal lobe that are more integrated, more connected in a way, also have connected temporal lobe as a whole in the entire brain network. And that last one, so the macro scale connectivity of the DMN has been linked to cognition in many, many different studies. And even in our study, verbal memory, which is the sort of parts of the DMN, was linked to the macroscale connectivity. So there are these crosslinks. It holds up. And for time's sake, I will skip this. Go to another perspective that you can have on resilience in the brain. The one I chose up to now was by only using static imaging. So we were trying to make every imaging modality integrate with the other imaging modalities in order to better explain cognition. This worked in a tracking way, in a monitoring way, but it didn't predict. But we can, of course, also look at network activities in a more dynamic way. So let me explain what I mean. So I showed you a short recording of an MEG, or it would also work for any other brain activity measurements. These are typically scans or recordings that take like 5 to 10 minutes. And up to now, everything I showed you is making a network of the nodes and the connections for the entire 5 to 10 minutes. So you have one network per patient, basically, or per participant. But 5 minutes is quite a long time. We know that there's a lot of in activity and also in network properties across time. So we can also analyze these recordings as consisting of multiple windows. And that's what we did in this study. So we focused here on the participation coefficient as a measure of centrality, which is the degree to which a node connects also to other nodes outside of its own module, which is illustrated here. The red node has the participation coefficient. And we did this on the whole scan. So then again, you have one matrix per subject. But we also looked at it from a dynamic perspective. So we divided the entire recording into epochs, which are really short bits of data, and then looked at the variation in this participation coefficient over time. Put the slide in, but we basically quantified how many times the participation coefficient really fluctuated from really high to really low. Thinking that if there's a lot of fluctuations, it must mean that the node is super adaptive to whatever is happening. So again, we had 2 time points for this. So we measured before the surgery and about 1 year after the surgery. And what we saw is the same type of sort of longitudinal correlation that we also found in the brain tumor patient. So on the x-axis, you see the dynamic connectivity measure. With a higher value indicating that there was more variability in whether nodes were connector hubs in this 5 to 10 minute scan. X-axis is the change in verbal memory in this case, so whether patients had declining verbal memory over time. You see some patients that did, so based on clinical cutoffs, these 5 red dots would be patients that decline significantly after the surgery. So those are the ones you really want to identify before surgery. Maybe not necessarily to immediately sort of change the strategy, but at least to give them some counseling on what to expect. And here we did find that the pre-operative, so the baseline dynamics of the brain, could predict the verbal outcomes. So here, it's not a change score on the x-axis, but it's the pre-operative value of dynamics of the brain predicting the outcome whether patients were to decline or not. And this adjusted R squared of 48% might sound very low. It's in brain behavior relationships, it's really, really high. We typically see more like 30%. So this is something that we really want to do on a much bigger data set and replicate. This gives some hope that we can sort of quantify this resilience for the surgeries. For what I want to close off on is what can we intervene? So I talked about using network theory and its application to the brain as a sort of predictor for whatever happens. Can we even specifically target sort of the brain network, knowing that some patients have issues in one part of the brain, others have issues in another part of the brain. And one way to do that is TMS, transcranial magnetic stimulation. It's completely non-invasive, so you see the brain here, but it's actually given outside of the skull. And you put this coil on top of the skull or scalp, and it delivers magnetic pulses, which induce action potentials. So you're basically directly either increasing the number of action potentials in the region underneath the TMS coil, or decreasing it depending on the frequency that you give the pulses with. And we know that this has, it has a very local effect. You can target it within 1 centimeter, the direct action potentials. But of course, the brain is super plastic. So as soon as you start doing this, and especially if you give repeated doses, we see changes throughout the entire brain in connectivity and activity. So it has a potentially global effect. Okay. Just want to show you what it looks like. So look as you see, you hear the click, which is the pulse. This is a single pulse, and for a moment, what one is looking at her left hand, which is like this, right? So we're waiting for her left hand to move because Lucas is stimulating her right motor cortex. So you would expect if he hits the correct region, the hand knob, that the left hand would move. So everyone is looking for that. I'm wondering why it's there.
Dat hij ook iets voelt, zoals hartslag.
Ja.
Als je dat doet, nu niet meer hoor. Maar...
Ja.
Ja.
Daar.
Oh, oké.
Oh, cool. Sorry.
And then you know that you're in the right location. So it's super small and it can be bigger.
Ja.
I did also, before you think that I had my PC students like zap only each other's brain, I also underwent it. And actually with me, they were in the wrong spot first. And then you put the volume up, so to say. So you keep giving stronger pulses. So by the time that they hit the correct spot, my whole arm is moving. So yeah, then you know that you're in the correct spot and what the general excitability is of the brain, because that differs per person and also depending on how much you slept or moving coffee, something like that. Once we know this, we can do it therapeutically. So now our question is, can we target the right location in each individual patient to maximize the therapeutic value of this treatment? This is a question we answered before for OCD. So those are patients with obsessive compulsive disorder, which means that they have very intrusive thoughts and compulsions that they are very hard to stop. There are some treatments for OCD, but there are still patients who benefit not so much from treatment. And then TMS is kind of being tested as a useful method. And what we did is this was a retrospective analysis of a study that already did the TMS based on anatomical location. So again, we target this frontoparietal network because cognitive control is part of depression, also OCD is relevant for the overall functioning of obsessions compulsions. But then we still observed that some patients did respond and others did not. So we used retrospective analysis to see whether we could predict who would benefit more. What we saw is that the greater TMS effect, so basically less resilience to the TMS, but more resilience on the sort of outcome of the study, was predicted by higher baseline connectivity. So if the target region in the brain was more connected in a sort of a local way, so it had a lot of connections within its own subnetwork of the brain, the frontoparietal network, then the TMS worked better. That's what you see here. We had a real TMS study, TMS arm, that's the sort of purple line, in which you see a clear correlation between the target, the connectivity of the target and the outcome. Whereas the active control, which was a stimulation that was not therapeutic, did not show the same correlation. So basically what we hope to do now, and we're doing a study in which we've already enrolled a couple of patients, is to use this to improve the cognitive complaints in glioma, knowing that they have both high clustering coefficient throughout the brain and also this low integration. And by targeting each individual patient with their own network, we hope that the effect is bigger. And you may wonder what are the side effects of TMS, because it is, of course, inducing action potentials. But the big fear is always epilepsy. The patients get seizures because you're kind of activating the brain from the outside. But that doesn't really seem to be a problem in glioma patients. So here you see a very preliminary study, feasibility study, where they did 5 days of TMS in glioma patients, and they didn't see any seizures. They did observe improvements in cognition. So this was not a controlled study. So we do expect all of these patients to improve during the period in which they were. But it at least tells us that it's safe to do TMS in glioma patients. And Maxine is doing a true gate study, which is actually trying to, in an RCT, improve executive functioning in these brain tumor patients. Let's skip this. So ultimately, what we hope is that we can better understand predict and hopefully treat these cognitive deficits in brain tumor patients. And we want to know more about the multi-scale work. This is a report that we wrote 2 years ago already. So on the other aspects that I didn't get to talk about because time restraints today. Thank you very much for your questions.
Thank you for this very fascinating talk. And I guess there's a lot of questions. So maybe I'll start.
Processing, yeah.
I mean, is it the topology of the network like it changes in time?
Yeah.
Right? And what is the processes doing governing that? I mean, why is that? And what time
Yeah.
scale is it happening?
It's happening on all time scales, I would say. So we have the time scale of a lifetime, where we see this inverted U shape. We see that on all levels. So, of course, there's a lot of change when you go from 0 to like 20. I think that's developmental, right? That's the aim to mature the brain. Then we have a semi-stable period for 30 years, maybe 40. Then it goes down again as people age, probably because of the limited viability of the body in general. So that's the longest time scale. But then on the shorter time scale, I think the brain networks change at the order of seconds, always. The question is, how big are the changes? So we do know that there's a large heritability of the brain network, especially if we look at the structural connections. Heritability of some brain regions is up to 0.8, which is the same as height. So that's very heritable. So that won't change in normal, healthy people that much. It's so encoded into our genome. The other parts of the brain are more variable. The first parietal network is one of those sort of sub parts of the brain network where there is a lot of variation depending on your environment, what you're learning, what type of exposure you have. And of course, if something goes wrong, brain diseases, so all that's wrong.
Okay.
Very nice talk, thank you.
Thanks.
At some point you connected the speed of the rise of the action potential to the connectivity or the centrality, as you call it, of the whole network. When I think of the action potential, I think of sort of a single neuron property rather than a network property. So how is that?
Well, that's the
linked?
interesting part. I think that's an assumption that we can challenge. I know that in the sort of basic neurosciences, every neuron is thought of as a singular unit, as a molecular or an atomic property or an atomic piece of the brain. I wonder whether that's really the case.
Okay, cool.
Yeah. So yeah, we only tested some neurons in each patient and then averaged over those. But it would be super interesting, of course, to look at bigger parts of the brain, to assess more neurons for each individual, and to have other brain regions. But we didn't have them because this was all temporal lobe sections. So we were limited by the experimental setup. But we're doing post-mortem work to look throughout the time.
That's very interesting. Thank you.
So, we have a good normal small worldness.
Yeah. Yeah, there's a lot of them actually. I didn't mention any of that in the talk, but there's a part of the community that's doing more generative modeling. So there you encode these different principles like a small worldness, but the one would be a preferential attachment, and it creates to some extent, to a reasonable extent, the development of the brain network. What we typically do in terms of modeling is to use, for instance, neural mass models to model individual brain regions, and we couple them according to the patient's own structural network and simulate functional brain activity as with neural mass model differential equations. And then the great thing is that we have experimental data to tune and validate our models, and we're actually also in a project trying to predict cognitive outcomes using these types of models instead of just measuring, which I think is the future ultimately.
I have one question. Because now for the patient is like after treatment, like the average like lifespan like 14 months. My question would be like, is there any method to like pre-diagnosis in such kind of tumor? For example, now use the brain network like to predict the cognitive ability after surgery. Can this kind of method also used to predict, for example, I myself, like diagnosis, for example, use a network, I check my own brain activity, and then, or probably I could have a tumor. Can this be used to early diagnosis?
Yeah, it's a very interesting question. The answer is I don't know, because we don't have measurements of people's brain network before they develop a tumor. But actually, I think this is very relevant, because what we do see is that brain tumors, especially the fast-growing tumors, they don't occur everywhere in the brain. They occur predominantly in the DMN, in the FPN, in the sort of hub type regions of the brain. Which mean, and that's how we know that, because we then compare the occurrence of the brain tumor with healthy people. But of course, that's assuming that the brain tumor patient was an average healthy person before they developed a brain tumor. That's a very reductionistic way of viewing brain disease, I think. And something that we can again challenge, because we see this link between kind of average healthy network topology and occurrence of tumors. Maybe some people just have a brain network that's more susceptible to brain tumors and other brain diseases, actually. So the interesting part there is that all sorts of brain related disorders are mostly found in the hubs of the brain. So this is not specific to glioma. We do see this vulnerability across different brain-related diseases. What we would need, and I've seen Alzheimer's disease, they're much more ahead of that, because of course, Alzheimer's is much more common. So there's a lot of cohort studies there, sort of including people when they are still relatively young and have no complaints, and then they follow 10,000 people and see which ones of them get Alzheimer's disease, and there the network is predictive. Yeah.
Question?
Thank you for a very interesting talk. I was wondering, in the end of your talk, you sort of mentioned this sort of targeted treatment method, and that it was somehow most effective on the more connected regions of the brain. Is this something that you would also target, if I flip it the other way around, so that you're looking at the most connected regions of the brain and you're sort of targeting specific nodes or connections in order to get to, let's say, a normal connectivity state, a normal module structure or something like that?
Yeah, that's the idea. We again challenge the idea that we have to stimulate to make people more normal. I feel like if you have a big tumor and a resection and all these issues, thinking that you can stimulate people back to normal is kind of useless. It will probably be some sort of a new normal, where some of the integration, which I think is key for this cognitive deficit, some of the integration is recovered. But you can't really go back to the same situation that normal person would have. And that's also not what we see if we look at the evidence of what happens after successful treatment on the behavioral scale. It's very hard to find consistent and similar patterns of change in individual patients. So some patients may come back, like be more like normal people after successful treatment. Others are more different from if the treatment is successful. And actually the baseline before the treatment is the most predictive of the behavioral outcome, not the change in the network, which is mystifying. Something we need to look at in much more detail. Yeah.
Thank you.
Thanks.
Okay, thank you very much.
Thank you.
So here's a small present from